Hello, hello, and welcome. Welcome to Chapter 8 of Concepts of Biology. Today we're going to be talking about patterns of inheritance. We're going to talk a little bit about Mendel and his experiments, as well as what happens when we see multiple alleles, multiple genes, all kinds of stuff. So let's get right into it. In this first section, we are going to explain some of the scientific reasons for why Mendel's experiments were so successful and why we still even reference and use them today. And we're also going to describe some of the expected outcomes of monohybrid crosses involving dominant and recessive alleles, and we will explain what all of those things are. But first, a little bit of introduction. This right here is Johann Gregor Mendel. We call him the father of genetics, of modern genetics at least. And he was a monk who studied pea plants. Johann. He studied pea plants and crossed them a million different times in a million different ways and discovered some really interesting facts about how these plants were getting both their seed color and the texture of their seeds. And we'll go into his experiments and what he did and why he did what he did. Okay? So while experimenting with thousands of garden pea plants, because remember, he was a monk, he was bored. When he wasn't praying, he was planting. He uncovered some of the fundamentals that we still understand and use today to explain genetic features. So part of Mendel's experimentation was using these garden pea plants to study inheritance. This species of pea plant does naturally self-fertilize. So the pollen on the ova can fertilize the plant itself, and it will grow new versions of this plant. In his studies, he maintained a pure colony of white flowers and a pure colony of purple flowers so that they did not mix. They did not cross-pollinate at all until he physically cross-pollinated them. We call these true breeding plants, or something that is true bred. It is purely one thing, okay? He then crossed those plants in a process that we call hybridization. And in that hybridization, he noticed that only the purple flowers of the parent generation were showing in that first generation of offspring. Okay. He then self-pollinated the F1 generation, this first generation of offspring, and he found that about 75% of the F2 generation ended up having those purple flowers, but about 25% had those white flowers that we saw disappear in our F1 generation. And that led him to question why, which led to all of the other types of genetic information that we now know and use today. I'll give you a little hint. It's the fact that it's a dominant gene. And we'll talk about what that means in just a few minutes. In his studies, Mendel identified 7 characteristics of pea plants that can be determined by kind of crossing those true bred plants. So the things that he looked for were, was the seed round or was it wrinkled? Was the seed yellow or was it green? Was the flower purple or was it white? Were the pods constricted or inflated? Were they green or were they yellow? Were the stems tall, short, or were the flowers at the top of the plant versus kind of towards the center? Those were the 7 traits that he identified. pea plant characteristics that he would use to prove his theory of genetic inheritance. Okay. So Mendel, in all of his scientific glory, has found that dominant traits are traits that are inherited. Inherited and are unchanged in a hybridization. Okay. So going back to our purple and white plants example, the purple plants did not change in that first round going from the parent generation to the F1 generation, right? Recessive traits become latent or, for lack of a better word, kind of turn off or disappear in the offspring of that hybridization. Turn off. But he did also find that that recessive trait can come back in the offspring of the offspring, so the F2 generation. Okay. So that's what we mean when we say a trait is a dominant trait versus a recessive trait. Okay. So now we're going to talk about some of the laws of inheritance and what that means and how we sort through that genetically. So we are going to explain the relationship between a genotype and a phenotype in the dominant and recessive gene systems. We're going to use Punnett squares to calculate the expected proportions of the genotypes and phenotypes in a monohybrid cross. We'll explain Mendel's law of segregation and independent assortment in terms of like genetics and meiosis and how all of that plays a role in this inheritance. And then we're going to explain the purpose and methods of a test cross, why we do it, what it shows us, all that jazz. Okay. So phenotypes and genotypes. Before we get too far into those phenotypes and genotypes, let's talk about alleles. An allele is just a different copy of the same gene. Okay. Every organism, every multicellular diploid organism specifically, has two copies of a gene, one from one parent and one from another parent. Alleles are what we call those different forms of that same gene. Like, for example, eye color. If one of your parents' genes codes for blue eyes and the other parent's genes code for brown eyes, I'll tell you right now, brown is the dominant trait. So that offspring will most likely show the brown eye trait. That's what we're talking about when we're talking alleles. Okay? When we are referring to these alleles, we use terms like homozygous dominant, homozygous recessive, or heterozygous. Homozygous dominant just means that the dominant form of both of those alleles is shown. Same with recessive. Heterozygous means that that person has one copy of the dominant allele and one copy of the recessive allele. And this is how we kind of notate them with those capital letters versus lowercase letters. Okay? The phenotype is the physical trait. It's what you can see. It is the specific eye color that you can see, right? It is observable or expressed by the organism. The genotype, however, is the genetic makeup. Of the individual. Okay. It has both the expressed and non-expressed alleles. Okay. Going back to our eye color example. If these two people had a kid and that kid showed up with brown eyes, their brown eyes would be the phenotype, but unless we did further genetic testing, we wouldn't know exactly what the genotype is, right? That kid could either have homozygous dominant brown eyes, he's going to have brown eyes no matter what, or he could have heterozygous brown eyes. We wouldn't know unless we tested his genome. The only thing that we would know is if he had blue eyes. That means that both of his parents gave him the recessive allele, which would in turn tell us that this parent, the one with blue eyes, is homozygous recessive, and this parent with the brown eyes is heterozygous. That's the only way that we would be able to get a child with blue eyes. If that didn't make sense to you, don't worry, we're going to get into it a little further. Okay? So like I was kind of saying, phenotype is the visual, physical trait that you can see. Genotype is the genetic makeup. They're not always going to be the same. Okay? In the phenotype, about 75% of those seeds ended up being yellow and only about 25% were green. But you don't know for sure, unless you've done the testing, that 25% are homozygous dominant for that yellow, 50% are heterozygous. That's kind of where that breakdown happens. Okay? This is another example of an allele here. This child here, cute little guy, has albinism. He is albino. The trait for albinism is recessive. That means both of his parents had at least one copy of the recessive gene. So his parents were probably AA and AA. And because of that, this child now has the possibility, I'm getting a little ahead of myself with the Punnett square, but this child now has the possibility of getting both of these recessive alleles and being AA. And that's what happened here with this little guy. I was just saying that I was getting a little bit ahead of myself with the Punnett square. Punnett squares are what biologists, geneticists use to determine the probable genetic and phenotypic ratios of an offspring. Okay? The probability is usually for a monohybrid cross if both parents are, well, it'll be different for whatever the parents are, but if both parents are Heterozygous, meaning both parents are big B, little b, big B, little b, the probability of their children's genes being homozygous dominant, homozygous recessive, or heterozygous are, let's actually flip that. We'll do heterozygous in the middle because it feels a little better. And homozygous recessive is about 1 to 2 to 1. That's the probability of what their genetic makeup would be, what their genotype would be. But their phenotype would probably be somewhere in the 3 to 1 range. Why? Because these two groups are going to show that dominant trait. Okay? Punnett squares are a mathematical measure of likelihood. It is the math behind why any of these things work. So let's run through one real quick. When you're making a Punnett square, what I like to do, you draw your little square, you make your little cross, and I put one parent on one side. For our example, we'll just use this guy here, or these two people here. One of them is, or they are both homozygous, sorry, they're both heterozygous, my bad. And then you just kind of fill it in as it goes. So one kid has the opportunity to be homozygous dominant. Two kids have the opportunity to be heterozygous. And you have about a 25% chance of getting a homozygous recessive kid. Just like we said. And then that phenotype, this one will show the dominant trait, this one will show the dominant trait, and so will this one. Punnett squares work. Don't know what to tell you. So when you're doing a monohybrid cross, that's kind of what we end up seeing. And this is that same kind of Punnett square with these pea plants. He used a homozygous dominant yellow plant, a homozygous recessive green plant. That offspring is only going to get a heterozygous plant, meaning that it will still show the dominant trait. Let's draw that one out real quick. And we're just going to use this example right here of the cross of the parent generation. So we have a Y and a Y and a little y and a little y. So now all of my offspring are going to be heterozygous They're all heterozygous. They're all going to show that yellow trait. If I were to cross one of my offspring, remember these plants are self-fertilizing, so if I cross it with itself, I'm going to get that 1 to 2 to 1 ratio. You know what I mean there? There. And then you can fill that out on your own if you want to prove it to yourself. So those are Punnett squares, and that's kind of how we use Punnett squares to determine the mathematical likelihood of a genotype or phenotype showing up in a population. Another thing that Mendel tried to kind of propose and that we still use today is the law of segregation. The actual definition is that this law states that paired unit factors or genes must segregate equally into gametes such that offspring have an equal likelihood of inheriting either factor. All that means is that during meiosis, each one gene, a pair of genes, because remember we start as a diploid 2N, at least one gene will go to each gamete. so that both offspring have the potential of gaining that trait, right? They will get the other pair from their other parent when that time comes. Okay. This law of segregation is based on that first division of meiosis, um, in which the homologous chromosomes separate into their daughter nuclei. Remember, we have homologous chromosomes that will then separate into their respective daughter cells and then get split even further, as is the process of meiosis. So each one of these offspring have the chance to get that gene that we are talking about, or that we are trying to pass on. And then this is kind of getting into that first division of meiosis that we were talking about. We remember those homologous chromosomes, they get pulled apart. We have our daughter cells, they continue on. So a test cross is a mechanism that we use to determine whether an organism that is expressing that dominant trait is homozygous dominant or is heterozygous. So if we don't know what the parent generation is, we can do a cross between a homozygous recessive and the trait in question, because you know that at least one of the alleles is showing that dominant trait. You might not know what the other one is, but you know at least one of them is dominant. From that offspring, you can see that if the offspring have some of the recessive trait, or if they are showing a recessive trait, you can assume that the parent is homozygous, is heterozygous. If you see that none of the offspring have any of that recessive trait, that tells you that the parent generation was homozygous dominant. So it's all in the testing of the offspring that you can tell what the parents were genetically. You can tell what their genotypes were. So we use test crosses to find out whether or not an organism is homozygous dominant or is heterozygous. So Mendel's law of independent assortment essentially just states that genes do not influence each other with regard to the sorting of alleles into gametes, and every possible combination of alleles for every gene is likely to occur. That's the scientific definition. But what does that actually mean? That means that during gamete formation, so all of the sex cell formation, gamete, during gamete formation, alleles of different genes sort independently of other genes. Alleles sort independently. Alleles Yeah. So during that gamete formation, alleles will sort without thinking about another gene. Like, for example, you know how girls tend to, and I'm not trying to girl shame right here, you know what I'm doing here. It's just an example. Girls tend to go to the bathroom together, right? So all of you and all your girls are going to the bathroom. One of your girls is just like, you know what? I don't need to pee. I'm okay. So she will independently sort. And that causes all of your other girls to be like, you know what? I actually don't need to go to the bathroom. I'm going to sit right here, or I'm going to go to the bar and get a drink, and I'm going to I'm going to go outside and make a call. Despite what all of the other genes are doing, each allele, in this example, each girl is going to go do whatever it is that she needs to do. That's essentially what that independent assortment is talking about. Okay. These two, for example, two traits like seed color and seed texture, they're going to sort however they want to sort. And this is all taking place in meiosis. Usually in that first division of meiosis, but it can come up in the second division. Okay, so that random segregation literally just means that you can get any combination of possible genetic arrangement. So because we are looking at two unrelated traits, we can do what we call a dihybrid cross. Before, we were doing monohybrid crosses, and that was only looking at one of the traits at a time. In a dihybrid cross, we're looking at two traits between the seed color and the seed texture. Okay, that's what we're looking at here. In dihybrid crosses, we find that the phenotype, remember phenotype is the physical trait, will generally be in a 9:3 to 3:1 ratio. So you have about 9 of them that are going to be homozygote or show at least the dominant trait for the color and dominant for the shape. 3 that are going to be recessive for color but dominant for shape. 3 that are going to be dominant for color but recessive for shape. And maybe one little guy that is recessive for both color and shape. Okay, so we do a dihybrid cross to show what the phenotypes might possibly be. We can't exactly know all of the genotypes without doing several different types of test crosses. So in this last little bit, we are going to talk about some of the extensions of those laws of inheritance, because remember, I always say in biology, there is no always, there is no never, there's always an exception. And of course, there are some exceptions. So we are going to identify some non-Mendelian inheritance patterns, such as incomplete dominance, codominance, multiple alleles, and sex linkages. We're going to explain the effect of linkage and recombination on those gametes genotypes. And we're also going to explain some of the phenotypic outcomes that we get from epistasis among different genes. And we'll talk about what epistasis is as well. So in incomplete dominance is where a heterozygote will show a phenotype that is kind of in the middle of both of the parents, right? So this snapdragon here is a heterozygous flower, meaning that it is big P, little p. It doesn't matter what the letter is. The letter can be whatever you decide for it to be. Or in their case, they decided it was red and white. So let's do that one, okay? So this guy is red and white. And so because he has both of these dominant traits, these technically dominant traits, they end up kind of sharing that dominance. So while the plant is not completely red, it is not completely white, it ended up being kind of a mix of those colors. And that's where you get that incomplete dominance, is where you get kind of a intermediate. Okay. Codominance is where both alleles for the same characteristics are simultaneously expressed in the heterozygote. Okay? That's different from incomplete dominance because in codominance, the parent allele is fully expressed. Both parental alleles are fully expressed separately. And that's why in things like blood type, you see that some people have AB blood, or I think that's the only AB. Yeah, AB blood. So they are getting both the complete expression of the type A blood and the complete expression of the type B blood. Okay, so that's where both of those alleles are completely expressed fully in that heterozygote offspring. Multiple alleles is pretty much exactly what it sounds like. It basically just states that a gene can sometimes have multiple different versions of the kind of same gene, more than two. So, for example, humans, the allele for blood type can either be A, B, or O. In some people, it'll be AB. In some people, it'll be BB. In some people, it'll be AO. Doesn't matter. They're all three versions, A, B, and O, come from the same gene. They're all just different alleles of the same gene. Sex-linked traits are traits that you will only find on an X or Y chromosome. They're only on a sex chromosome. For males, these sex chromosomes are an X and a Y. And for females, it is two X's. Okay. If a gene is located on the X or Y chromosome, we call it a sex-linked gene. Okay. And it very well could be X-linked. A lot of the sex-linked traits are X-linked, meaning that they will show up on the X chromosome. Okay. For example, eye color in Drosophila, which are fruit flies, is a sex-linked trait. Okay. In humans, an example of a sex-linked trait could be hemophilia, which is the inability to clot blood. Or color blindness. You'll find that color blindness is generally usually found in males because they don't have another X to try to fix it. It's a whole thing. We'll talk about it later. Okay. So, like I was saying, that eye color in these fruit flies, the red eye color is wild type, so is dominant. Wild type usually just means dominant, maybe not even dominant, but what you would see in nature most commonly. To the white eye color. So this guy has the dominant gene. This guy has the recessive. When we do Punnett squares based on X-linked genes, you got to be a little bit more careful because we are using both an X and an X and an X and a Y. So it's just kind of making sure that you have your traits in the right places. Say that, so going with this example, the female is homozygous dominant, I think is what I'm seeing there. It's a little blurry, my bad. And the male, since he only has one X, he only gets that trait put on one X. And he is homozygous recessive. So what are the chances that their male child will have white eyes? We don't know. Well, You guys do the Punnett square, see if you can figure it out. Oh, I didn't read that, sorry. In the diagram, the little W is the white mutant eyes, and I think Mom has at least one of those little W's. So let's do that, and let's do that. Give that cross a try and see what you get. So, linked genes. Didn't we just say that genes sort independently of one another, and now we're talking about genes being linked? Yeah, I told you. There's always exceptions in biology. Um, so the segregation of these alleles can be influenced by linkage, and all that means is that genes that are actually physically located next to each other are more likely to be inherited as a pair, right? So if you're looking at your genome and you've got like you got a gene for whatever, and you got another gene literally right next to it, there's a high likelihood that these two genes are going to, when or if they do get translocated or anything like, or moved to another chromosome, there's a high likelihood that they're going to go together just because they are physically closer to each other. But the process of recombination and crossing over means that it is possible to have two genes on the same chromosome behave independently or if they were not linked. So it's possible, but a lot of the times, the closer they are physically to one another, the more likely they will be inherited as a pair. Okay? So in this process of crossing over, right, we talked about this a little bit in meiosis and mitosis, you get some recombinant chromosomes. A lot of the times, what we find is that a single observable characteristic, like, for example, the color of your eyes, the or how tall you are, something like that, right? That tends to be under the influence of many, many, many genes. It's not just one gene that controls all of eye color. It's, for example, at least 8 genes in humans will contribute to eye color. So things like your height will have several things that are contributed to it. The color of your skin, lots of different genes will contribute to it. Okay? There is a process that we call epistasis, in which some genes will work to kind of cancel or mask the expression of another. And that happens all the time. Some genes will be there and active, and there will be another gene later down the line in another chromosome that will completely counteract the effects of all of those chromosomes. That's why we have some differences. Even brown eyes have some differences in how they present. Green eyes have differences in how they present, and so on and so forth. No 3 blue eyes are the same, no 3 green eyes are the same, and no 3 brown eyes are the same. It's that epistasis, that interaction of genes, that's playing a role in kind of seeing whether or not a trait will be fully expressed. Same thing with skin tone. So in this particular example of epistasis, the gene for coat color may be the C gene, but you don't see any coat color unless the A gene is expressed. That's what it's saying here. So when you have expression of that A allele, you do get a coat color. If you have expression of the A allele and no expression of the C allele, you'll get an albino mouse. And that's kind of what that epistasis is just showing.